Hello, thanks for coming, everyone. It's my pleasure to introduce our speaker for this great edition of the Neuroscience Talk Series. Dr. SILVIA Pellegrini is here today, and she's not a neuroscientist or a psychologist by training. So why is she here? She is an associate professor of clinical biochemistry and clinical molecular biology at the University of Pisa, where she got her doctoral degree in experimental oncology and tumor morphology. And she tells me that she's changed her research direction. So now she's studying the molecular biological basis, the genetic basis of behavior, and doing some really fascinating studies of the genetics underlying some extreme aggressive behavior. And so I think I'm looking forward to this talk as much as anybody else in this room. And I will let you talk.
Okay. Okay, thank you, Jim. I'm very happy to be here. And first of all, I like to thank Professor Jim Axby for inviting me to give this talk. And as he told, I'm a professor of clinical biochemistry and molecular biology at the University of Pisa in Italy. And my research activity is mostly focused on the social behavior, the molecular basis of social behavior. And that behavioral traits do have some genetic component has long been known by observational studies in twins and adoptees. Adoptive children and parents, in fact, share the same environment, but not genetics. And at the opposite, monozygotic twins raised apart have identical genomes, but different environmental stimuli. And heterozygotic twins, as well as siblings, represent an intermediate situation because they share 50% of their genomes and the environment. And based on this kind of studies, observational studies on twins and adoptees, we now know that, for example, for each big 5 traits, the correlation for monozygotic twins are higher than for dizygotic twins. And we also know that around 40% and sometimes over 40% of the variance for each trait is explained by genetics. And it's also known that even the IQ is positively correlated to the number of shared genes. So monozygotic twins share a higher number of genes, and they are more similar also in terms of IQ. And thus, if the genetic profile affects all the aspects of normal and pathological human behavior, how does it do it? It's evident that people differ from each other, not only concerning their height, weight, eye or hair color, but also for the way they behave. And most of these differences depends on their DNA. And each subject indeed has his own genome sequence, and the genomes of 2 randomly selected individuals differ for at least 3 billions of nucleotides, 3 billions of variants, of genetic variants. Only identical twins share the same genomes. But actually, also the resemblance between monozygotic twins is never absolute. And these differences increase during their lifetime. So probably the environments play an important role in making this difference. And this happens because they have been exposed to different environments that affect gene expressions without modifying the DNA sequence, what is now known as epigenetics. And this fact explains also why even behavior derives from a strict interplay between interaction between genes and environment. And after, this is true also for aggressive behavior. It's widely known, it's widely believed, for example, that violent behavior is the result of a combination of genetics and environment, and with the two factors playing similar roles, equal roles. As a matter of fact, after the decoding of the human genome sequence, we now have the instruments to study, to investigate the association between some specific genetic variants and behavioral traits, including aggressive behavior. The first and the most famous gene reported as associated with abnormal behavior is the MAOA gene that codifies for the monoamino oxidase gene, monoamino oxidase A. This is an enzyme. And this gene is also called the warrior gene because of its role. And MAOA codes for monoamino oxidase, an enzyme that metabolize monoamine neurotransmitters released in the central nervous systems. So the first association between MAOA and aggressive behavior dates back in 1993, when Brunner and colleagues reported the case of a Dutch family in which most of the male members were particularly violent, particularly aggressive. They committed violent crimes, homicides, rapes, arsonism, and very interestingly, these individuals, all the affected individuals, the male individuals, had a stop codon, a mutation, a genetic variant, a stop codon in the MAOA gene that resulted in a completely ineffective enzyme. That means that just a single of one nucleotide make a stop codon in the reading of the sequence that make a truncated enzyme that was completely ineffective. And the MAOA gene is located on the X chromosome, and this is why only males were affected by this behavior. This was the first evidence of relationships between MAOA enzyme and aggressive behavior. And this one was the only evidence that there was a deterministic effect between a MAOA, a genetic difference, a genetic variant, the one in the MAOA, and behavior, aggressive behavior. But it's the only example that we know till now, because this mutation, the dramatic stop codon variant described by Brunner, has never been found outside the Dutch family. Okay, it was a mutation exclusive of that family, and fortunately maybe, but while another mutation, another variant exists in the sequence of MAOA, in the promoter sequence of these genes, that is pretty common in the general population. Its frequency is around 3.5% of people with the low allele variant, and that results in the synthesis of a less effective enzyme. So people with the low variants, the low allele of these variants, they do have an enzyme, a functioning enzyme, but this enzyme is less effective than people with a high allele of MAOA. So it's different from the one in the Dutch family that I showed you before, because here people didn't have any functioning enzyme, MAOA enzyme. In this case, people with the low variant have a less functioning enzyme, is less ineffective enzyme. And these variants have been named MAOA low, in contrast to the wild type MAOA high, to indicate the different enzyme efficiency. Caspi and co-workers at the beginning of the 2000s showed that individuals with the MAOA low exposed to negative environments during childhood had a significantly greater probability to develop antisocial behavior as adults as compared to individuals with a high variance of MAOA. Interestingly, in the absence of negative childhood environment, there were no difference between in the probability to develop antisocial behavior between people with a low variance and a high variance. This was the first demonstration for the first time. The results from the CASP studies that represent the starting of this kind of studies demonstrate that a strict interplay between genes and environment increase the risk of developing aggressive behavior as adults. While the effects on behavior of the MAOA gene is a relatively recent acquisition, our group found a MAOA low variant also in the Giovanni Dalle Bande Nere. Giovanni Dalle Bande Nere was a troop leader, an ancient troop leader, a condottiero in the Renaissance in Italy in the 16th century. He was notorious for his particularly violent behavior. It suffices to say that he committed his first homicide when he was 13. And he committed many other violent acts during his short life. He died as a young, he was 33, after an injury in a battle. And he was also raised in a very negative environment because his father was Cosimo de' Medici, and he died a few months after the birth of Giovanni. And his mother, Caterina Sforza, also known as the warrior lady of the Renaissance, was spent her life in fighting enemies, mostly Cesare Borgia, to defend their properties from the enemies. So I think that probably she wasn't a very affecting mother, present mother, war mother, as we can imagine. So a very negative environment, and we in collaboration with the paleopathologists of our faculty in Pisa, we obtained a little bone fragment, a femur fragment of Giovanni Dalle Bande Nere. We extract DNA and we found a low allele of MAOA. That probably our hypothesis was that in combination with the very neglected environment where he grew up, probably determine or affect his violent behavior. And MAOA is not the only one gene linked to aggressive behavior, linked to violence. Other genetic variants have been associated in scientific literature with the aggressive behavior. For example, the 5-HTTLPR variant that is located in the promoter of the serotonin transporter gene. This variant, the short variant, decreases the expression of the gene, thus reducing the availability of the transporter and increasing the permanence of the serotonin in the synaptic cleft. So this variant, the short variant of the 5-HTTLPR, has been associated with reduced impulse control, with increased anxiety, and sometimes also with depression. And other variants known to be associated with aggressive behavior are, for example, variants located on the dopamine D4 receptor. We know two variants. One is a VNTR, this is a variable number of tandem repeat variants. And the other one is a SNP variant in the dopamine receptor D4, both affecting gene expression and consequently the receptor efficiency. They have been linked to sensation-seeking behavior and to addiction. And another one is the emissions variant in the catechol-methyltransferase, that is the other enzyme that metabolizes neurotransmitters in addition to MAOA. And this variant produces an amino acid change that results in a less efficient enzyme. So more than one genetic variant has been today associated to aggressive behavior in the scientific literature. And, but all these findings have any relevance from a forensic law perspective? I mean, should we consider criminals bad as they freely choose their actions, or we should consider them mad as they couldn't act differently? This is the question that we try to answer. And as a few years ago, the genetic background of a defendant who carried all the variants of that increased risk, as you can see here, the red one are the risk variant, the risk alleles linked of these genes linked to aggressive behavior in the scientific literature. So this defendant carried all the variants of increased risk, and was his genetic background was among the additional neuroscientific evidence that was taken into consideration by an Italian judge to reduce by 1 year the penalty of a killer because of partial mental insanity. Okay, so this case was in 2009, and the news of this event went around all over the world. There were really hundreds of files on Google talking about this case, and you can see the Times, Le Monde, and other German papers, and it came also in Nature, and it was also discussed in Nature as the first case of genetic evidence accepted by a court reported as lighter sentence for murder with bad genes. Okay, so this was the way in which this sentence was received, but actually this is not the right interpretation of the sentence motivation. So the experts never say that the cause of the behavior of this guy was his genetic profile. But the two experts appointed by the judge, one is here, this is Professor Pietrini, the other one is Professor Sartori in the University of Padua, wrote a report that included a complete psychiatric evaluation, neuropsychological test results, and in addition, brain magnetic resonance, both structural and functional magnetic resonance, and genetics. And thus they didn't reach their conclusion on the partial mental insanity just because this guy had bad genes. Rather, the additional neuroscientific studies were used to obtain a clearer picture of the motivation. So it's different from saying that the genetic was the cause of the action of this guy. It's really different. And in 2011, a second sentence by another Italian judge, this was in Como, was the court of Como, okay, was based on an expert report that also included results from MRI and genetic studies. And the experts were also, in this case, Professor Pietrini and Professor Sartori. But in this case, the sentence was reduced, the penalty was reduced from 30 to 20 years, anticipated by a period of 3 years in a mental hospital. Just to say you something more, this was a case in which a young girl killed her sister and then burned her body, the body of the sister, and tried to kill also her parents by burning them. But the police were able to stop her while she's trying to kill her mother. And she has a bad genetics in terms of risk alleles. This is her profile. And the important thing that he was completely unaware of the bad things that he had committed. So the 3 years that anticipated the penalty in the prison probably make her conscious, more conscious that what she did. And in 2011, in the same year, also in the United States, there was a case in which the Court of the Appeal of Tennessee changed from that to life sentence because the defendant had the MAOA law allele. So this was a different case in the United States. But to date, we have collected genetic data from over 30 individuals. All these individuals came to the attention of Professor Pietrini, Professor Sartori, or other experts as a criminal case. And all these people committed impulsive crimes, mostly homicides. This table shows you the genetic profile of these people concerning the variants that I told you before, genetic variants that I told. And as you can see, in red, I reported all the risk variants of the literature. So you can say that all these people have at least one or both the allele for each of these genetic variants that we considered. So all the criminals have several high risk variants. But, sorry.
So how frequent are these kind of genetic profiles in just a random selection of people who do not have aggressive histories?
We're not good. Okay, I'll show you.
Okay.
I'll show you. Yes. In fact, by comparing criminals to controls, so you anticipated my talk. Thank you. Okay, by control subject with no history of criminal behavior or violent behavior, we observed that criminals have a greater number of high-risk variants as compared to controls. And in red you can see criminals, in blue you can see the controls. And in particular, only a few controls have 5 variants, and no one has 6 variants. Okay, so each variants independently are in similar distribution in the 2 populations, except for MAOA, DRD4, UVNTR that are more frequent in criminals. But if you combine together all the variants, the difference is more clear. Then criminals have a combining profile more frequently than control subjects. And anyway, 30 people, there is a small number to reach any statistically significant results. We are aware of it, and this is the reason why we didn't publish these results yet. But at the beginning of this year, we established a collaborative project with Professor Kent Keel of the University of New Mexico. He's famous in the United States because he put an MRI machine on a truck, and he went around all over the prison in the United States, scanning inmates and collecting psychometric data. And also, he collected also salivas, and he gave me around 2,000 salivas from these people, from these criminals, to make the genetic study. So we just started to analyze this data, to collect genetic information, but we hope that we can finally reach the statistical significance to validate our hypothesis. And from the whole sample of 2,000 subjects that Professor Keel gave us, we firstly selected 539 subjects because they were white males, because for the genetic studies, the very big problem is the ethnicity. So we need a sample that is more uniform in terms of ethnicity. So we decided to start from this sample, this subsample, white males, because it was the bigger one inside of the whole sample. And they were all white males, older than 18 years. They were homogeneous for ethnicity, and we started genotyped 3 variants belonging to the serotonin pathway. We genotyped the 5 HTTLPR, we genotype the MAOA variants, and we also add another variant, a SNP on the HTLPR1B. HTLPR1B acts both as a presynaptic and postsynaptic receptor, and is known as CT change, a nucleotide change SNP. In its sequence, it was T allele. The SNPs is the one I report in the slide, and the T allele of this change interacts with a microRNA, and this phenomenon reduces the expression of the receptor. So the consequence is a reduced availability of the receptors, and probably an increased serotonin availability, because the reduced expression of the receptor cause the decreasing of the inhibitory effect, the inhibitory feedback that usually this receptor play as a presynaptic receptor. And it has been associated, this variant, also with conduct disorders, impulse discontrol, with greater aggression in rodents. So we decided to consider also these variants, also because we obtained some significant association within the TT genotype of this variant, of the HTLPR1B variant, and Eiger score of PCLR20, that's known as a scale that is useful to measure psychopath traits, and in particular to the Eiger score to the PCLR factor 2, that is known to be related to antisocial personality disorder, social deviance, sensation seeking, impulse discontrol, high risk of suicide, and so on and so forth. So we decided to add this variant to the other two that I mentioned before, the 5-HTTLPR and the MAOA. And we calculate, based on the functional data present in the scientific literature, a global risk score of developing aggressive behavior. I mean, as you can see there, the higher risk score, higher risk variants, are that variant that increase the availability of serotonin, and the low risk variants are the one that decrease the availability of serotonin. So we add each genotype to each other to obtain, to calculate a score of impulsive behavior, of aggressive behavior. A score that is predicting of development of aggressive behavior. You can also see here the distribution of all these genotypes based on their score in the criminal population. And in collaboration with groups of Professor Pietrini at the IMT School for Advanced Studies in Lucca, we perform a correlation analysis between genetics and structural MRI data coming from the scanning acquisition from Professor Kent Kiel, and by applying multiple linear regression model, including all the variables that you can see reported here. And now I will show some preliminary data, because just we obtained this data this week, but they are promising, so I like to show you what we found. So first of all, we found a negative correlation between the serotonin genetic risk score and the overall cortical volume. That means that the higher is the risk profile, the risk serotonin profile, genetic profile, the smaller was the overall cortical volume. As you can see, yes, there is a significance between HTT score and cortical volume. So we wondered which area was important for this aspect, and we decided to repeat the analysis by subdividing for the 5 cortical lobes, and we found a significant negative correlation with the right limbic lobe volume. So not only overall the global volume, but specifically with the limbic area, the limbic lobe, and you can see here the negative correlation. And we finally take into account the 5 limbic regions. So we found a significant correlation, negative correlation, with the rostral anterior cingulate cortex. You can see here. And these findings show that the genetic profile may affect the volume of a limbic region, the rostral anterior cingulate cortex, that is known to be involved in other functions like attention allocation, reward anticipation, decision making, ethics and morality, and also impulse control, discontrol, and emotion. So as I say before, this was just some preliminary data, and but I think that it could be exciting. So it's a good point to start with all the whole sample. But genes are not all. This is important. This is I'd like to be that one as the take-home message of today's talk, because these genes are not all. The environment is still crucial, and now we know that a specific genotype in association with a very bad environment, negative environment experienced during childhood, increase the probability of developing aggressive behavior. Okay, so but what happens when the environment is protective? At the opposite, not a negative environment, a very positive and protective environment, something like this. The same genetic profile. Okay, exactly the same genetic profile, the same genetic variants that I showed before as risk alleles for aggressive behavior, but in combination with a very positive environment. So what happens? What could you expect? In the graphic that I showed before from the CASPI work, we could already observe that there is an inversion of the two curves when environment is positive. Okay, so probably people carrying the low MAOA having reduced probability to become antisocial compared to people with a high MAOA allele. I mean, when the environment is very bad, is very negative, people with low variance at a higher, probably significant higher probability to become antisocial. But if the environment is not so negative, it's at the opposite, it's positive, the two curves are inverted. So maybe people with a low variance have a less probability to become antisocial compared to people with a high variance of MAOA. You can see here. And thus, rather, the concept has now shifted from genes, genetic vulnerability to aggressive behavior, to genetic plasticity. Maybe specifically, the higher is the number of plasticity variants, the greater are the effects of either negative, as the red arrow shows you here, or positive environmental factor. So the red arrows indicate people with the higher number of risk alleles, and you can see that both if the environment is very negative or very positive, we can have the opposite outcomes. On the contrary, individuals who don't have this variance, those indicated by the red arrows in this picture, these people probably are more resistant to environmental factors, both the negative and to the positive ones. Put in other words, we can say that this variance may be considered to increase the risk of developing aggressive behavior if the individual has been exposed to negative environment, but this was what initial research was focused on, but they probably make people more sensitive also to the positive environment. So their probability to become antisocial is reduced compared to people with the other variants. That is, this genetic variance increase the permeability, the sensitivity of the individual to the environmental factors. On the contrary, individuals who don't have this variant, this one, are more resistant to environmental factors, both to the negative and to the positive ones. Thus, what was believed in the past, that nature and nurture diverge from each other, is not more true. Nature and nurture meet each other to determine phenotype, and this is true also for aggressive behavior. We just published this mini review on the epigenetics basis of aggressive behavior. And to conclude, that what we have learned by studying genes in relation to behavior is that the genes have no deterministic effect on behavior. This is important. This is the take-home message for me. Talking about genetic risk variance may be reductive at this point, and it's more correct to talk about plasticity genes that make carriers more susceptible to both negative and positive environmental stimuli. And I like to say that these studies highlight the importance of the environment, both from an educative and a rehabilitative perspective. I'd like to thank people in my lab that works on this project, Sara Palumbo and Veronica Mariotti, and all the other collaborators, the groups of Professor Pietrini at the IMT, and Ken Kiel and Karl Arensky that gave us the saliva for criminals, so they represent the very starting point for our research. Thank you for your attention. If you have any questions.
I'm always been curious with the language we choose to talk about how genes and the environment interact. One of the things I've recently become curious about is that when we talk about the interaction taking place, I often wonder about how that interaction really functions. In this example, is it that the environment turns on aggressive behavior, like throwing a switch, or is it turn up the volume? What is it actually when we say that they interact with? What is it that we're really saying about that? How does that interaction take place? Is it some sort of increase in dimension, or is it just a switch that gets thrown on? Do you have any thoughts about that, or does that make sense?
No, you mean how genes and the environment interacts together to make these results, to make these the phenotypes? Yes. This is just as observational studies, so we can observe that there is a different frequency of genetic in the two groups of people that we compared, and our hypothesis, sustained by some scientific literature, is going toward the idea that the genetic profiles combining with the environment is the cause of the results. But probably we can suppose also that the environment can modulate the expression of the genes by some epigenetic mechanism, and the presence of the genetic profile, I mean the genetic variants, make difference in responding to the environment and to make the epigenetic modification of the DNA sequence, I think. Something like this. Okay.
I'll ask the important question.
I'll ask the dumb question.
We know that alcohol, let's say, it affects some people, calms them down, and others become aggressive. Mm-hmm. What is interaction? Is it related anyhow to those genes? Let's say people could live normal life without any aggressive behavior. They drink, they become violent, right? And other people, they could just have a drink and they, just fall under the table and they're fine, right? So, did anybody study this? And kind of a related question: do we have profile across countries? We know that Dutch people are dangerous now, right? But like different maybe nationalities or have different predispositions. Do we know that from big genetic surveys?
Different nationality? I mean, different.
Well, different because we talk about genes, right? So it's probably nationality is not just place of living. Right? I mean, ethnicities probably would be the better term. Yes.
Yes, I think ethnicity is a very important in genetic studies because it is a very big confounding factors. When you compare different people, different groups, if they have different ethnicity, you can find some differences that is caused by ethnicity and not by the phenotype that you're looking for. But I don't know, obviously, but the very good thing would be to find something that is cross-related in different ethnicity, I mean, at the genetic basis. Then the environment, different environment can make the difference. So what you say about alcohol or the people that can resist better than others, and maybe it is also related to other genes, not just neurotransmitters, but alcohol dehydrogenase and other enzymes, that's me. But
yeah, I mean, it depends on one with whom I go drinking, so I know what to expect.
Yes, but the different genetic makeup could make the difference, at least in this case.
So you've identified this profile for aggressive behavior with these 5 alleles.
Yeah.
Is there anything comparable? Are there a set of alleles that influence intelligence, or a set of alleles that can influence proneness to anxiety or introversion?
Yeah.
If so, do you think you get the same kind of environment by genes interaction, where there's a sort of just a sensitivity to the environment for intellectual growth that people with these plasticity genes would benefit from, or if it would, in a more impoverished environment, they would suffer more than people who don't have them. So, do you think this is like a general principle for behavioral genetics?
I think so, yes. Would be the basis of the behavioral genetics, I think so. Why not? Different phenotypes, maybe not this specific profile, maybe other genetic profile, maybe some of these alleles are involved also in developing intellectual abilities, and so yes, having a different genetic profile maybe make everyone differently sensitive to the environment also in these terms. Yes, I think so. Yeah.
I had a question about therapeutic intervention for people with different genetic profiles. I was wondering, when you have this flat graph that if you don't have any of the 5 alleles, the environment doesn't affect you anymore. And that if this is a general principle for neurological or psychological disorders, are there a subset of people that therapy will just never help at all? And do we need to rethink like what intervention should be for those kinds of people?
Yes, I don't know. There are no data now about this, but I think that yes, the hypothesis is that people with this kind of profile, genetic profile, probably are more sensitive also to therapeutic intervention, mostly behavioral intervention. Yes, I think so.
I'm wondering if you're planning to look at any epistatic effects. So, like gene-gene interactions, right? Because in your score, it looks like you're just putting them all into one factor. So I'm wondering if you could get even more information by putting your 3 genotypes in as different factors and look for even bigger effect sizes. Because it seems like that's, with all these GWAS studies coming up negative, it seems like a lot of the even big effects in maybe the general population and maybe in these populations could be gene-gene interactions rather than single gene.
Yeah, yes. I mean, these are just 5 alleles that are the best candidates to have a role in these phenotypes, but probably many other alleles are. But genome-wides are very potential, but I think that they didn't give the result as we thought before because of the power. Because if you look at all the alleles in the genomes, we really need 1,000 and 1,000 people. So, at my knowledge, there are no very exciting results from genome-wides till now. I prefer candidate genes because there is an idea, there is an hypothesis, and so you can. if the hypothesis is wrong, it's wrong. So you can find anything. But I think that is more promising candidate genes than genome-wide studies.
So that raises the question of how do you find candidate genes? Say your interest is in musicality, right? Who has genetic predisposition to be very musical, and who's going to be tone deaf no matter what you do, right?
Yeah.
But how do you if you have that question, how do you start to look for genes that might be associated with amusia or musical genius?
Good question. But usually you start from the literature. So if someone else has found association with this particular phenotype, or maybe based on the functional data about the genes. So if they increase in the specific case the serotonin availability. So I don't know.
Well, you showed us data of a pedigree from families, right? You do classical genetics, where you have familial inheritance.
Yeah.
So if you get lucky with a family, that might produce a gene, a candidate.
Yes, but
just do classical.
Yes, but that was the linkage studies. It was completely different because there was a causative genes, a stop codon, and when people have that mutation, they had the phenotype. This is rare, very rare. Usually they have most genes and most environmental factors that play together. So there are multifactorial phenotypes. So the typical study, the typical pedigree like this is not. It doesn't function with this kind of study.
But you could use those extremes to then identify genes to get to Jim's point, and then do like exome sequencing to find the SNPs that might be common in your general population.
Right.
So most of us believe in free will and believe in the penal system, and therefore it's comforting to hear that no matter what your genetics are, you have a choice of the environment, so forth and so on. But I wonder whether you can really conclude that from the data. The difficulty is that you've only begun really to scratch the surface of the genetic effect on tendency toward violence, right? You have identified a few genes, but as was pointed out here, that the interactions between them could be more complicated. There's likely hundreds of other genes that play a role. And when we know more, there's nothing in this data that would suggest that there won't be some combinations of genes for which it's hopeless. Right? And then when we find that out, then the courts really have a problem, right? And this could happen. There's right now we just don't have the data. But we only have rough stuff, and the rough stuff is never going to tell us that the genetic thing is absolute, because we just don't have absolute genetics.
Yes, I don't think that it could never be in just a genetic matter. Also, if you add other genes, maybe the environment is playing an important role.
But even with risk genes, it's the relationship between the gene and the environment. So, for example, that serotonin transporter gene she was talking about, if you're exposed when you're young to a tough environment, it puts you at risk for PTSD, but protects you the other way. Like, it depends on your childhood plus that gene. So I don't think you're ever going to be able to do that determinism, because what it does is the gene makes you more, depending on which transporter gene you have, it makes you more or less reactive to the environment.
Right, but we know that for some genes, but there are other genes that we know are very deterministic.
Yeah.
And we may not have discovered the ones that affect violence.
But it's unlikely you're going to line up your entire genotype of deterministic genes, although it's possible.
If I may intervene on this, since I've been the consultant for the cases that Silvia mentioned for the judge in 2009 and 2011, if I may. The issue here is that, in terms of criminogenetics and criminogenesis and criminodynamic, So what you see in courts usually is that if you have a different consultant, one for appointed by the judge, so the judge expert, one by the attorney general, then the defendant, and the victim, is like the old Jewish proverb which says if you have two Jewish people in a room, you have at least 3 opinions. It's exactly the same. So if you have 5 different opinions, if you have 4 consultants, and they are very assertive. So unlike the other branches of medicine, psychiatry, and forensic psychiatry in particular, is very subjective. I mean, you wouldn't say in medicine that patient is diabetic because I say so, and I am a great clinician. You say that patient is diabetic because his glycemia is over 140 milligrams per 100 milliliters in fasting, and then is what you find here is that you say that person is diabetic because I say so, or even worse, and this happened no later than 10 days ago in a case I was doing in Genova of a guy who killed his girlfriend out of. He said, yes, he has all these traits, but they're just traits, are not. So it's like saying, yes, he has a high glycemia, but he has high glycemia. He's not diabetic. He has high glycemia. If you have high glycemia, you're diabetic. So in medicines, this is an objective criteria. In psychiatry, in forensic psychiatry, there is this thing. So the aim of this is, can we find objective measures that can reduce the degree of variability that you have when you examine a person, so that, clinically speaking, the concordance rate among psychiatrists is 55%, which means if you take a quarter and you launch a quarter, after it's basically the same thing, 55%. In forensic psychiatry, you get even worse. So what people do usually is I want him to be insane, I want him to be sane or fully capable, and then I fit reality around that. So what are diagnostic criteria becomes traits that everybody has. The most common thing when we want to criticize, for example, abnormality you see on the brain by doing MRI scans is, this in court. If all people who are here now, we get to an MRI scan, I do find something on you by chance, which is not true. And which is may be true sometimes, but it must be a causal link. And if you find something in the frontal lobes of a patient, that could be relevant. So the working hypothesis is, can we come up with something that I called a functional frontal fragility syndrome, where you have a syndrome in the Greek term, so syndra, different things that run together in different combinations. It could be a genetic vulnerability, could be cognitive limitation, could be functional or structural brain abnormalities, prolonged stress, sleep deprivation. We have done studies on that. Sleep deprivation leads to local sleep in executive areas, bad environment. You combine all this, and then you get that somebody is more vulnerable to release of acting out or explosive behavior or impulsive behavior. Then, this scientifically. Then, from a court point of view, that is free will. Can we measure free will the same way we measure glycemia? This is what when I started my course at university, I said, the challenge is, will we ever get to measure free will the same way we can measure glycemia? It's a provocative thing, but it's the way I want students to think during the course. Then you can say, if you are a judge, you can say, well, who cares? By definition, this is natural variability, so I don't care if somebody has a genetic vulnerability to be more impulsive, to be less able to control impulses. This is business, because this is genetic variability. Or you can take that into account. And especially in terms of rehabilitation, you can think of, this is the same way, 150 years ago, we thought of epileptic guys like they were taken by evil, and the remedy was to throw them down the hill. Now we know that epilepsy is a banal neurological disorder, very fastidious, but because neurons discharge when they want. And if you have these neurons discharging in the temporal lobes, you do very strange things, for example. So to what extent do we have the ability to measure free will in impulsive discontrol? impulsive behavior. This is the working idea.
You shut down everything.
I don't want to be.
Well, thank you very much.
You're welcome. Thank you.